Thank you for giving me the opportunity to present my research at this forum. Today, I will be presenting on the relatedness of global populations of a very devastating Eucalyptus foliar pathogen called Teratosphaeria destructans. Teratosphaeria destructans causes severe shoot and leaf blight, which leads to the defoliation and dieback of newly planted eucalyptus trees, especially those cultivated outside of their native environment. Asexual fruiting structures form below the stomata under moist conditions, and they exude these asexual spores, which gets dispersed to the neighboring trees. The pathogen was first discovered and described from North Sumatra, Indonesia in 1995, after which it rapidly spread to other tropical and subtropical eucalyptus growing regions around the world. This was most likely with contaminated seeds. It was reported in Thailand in 1999, in East Timor and Vietnam in 2002, in China in 2004, in Laos in 2009, and for the first time outside of Southeast Asia, it was reported to cause severe devastation in the eucalyptus plantations in South Africa in 2015. And our most recent report of this pathogen causing disease was found in Malaysia in 2016. And as you can see, this pathogen is restricted at the moment to areas in Southeast Asia as well as South Africa. And even though this pathogen was first discovered in 1995, very little is known regarding its biology or pathology. Therefore, the aim of the study was to investigate the genetic diversity, the potential for recombination, and the global relatedness of this pathogen of 7 populations. So we sequenced 3 genomes, and we used these genomes to develop 10 polymorphic microsatellite markers. These markers were used to assess a global collection of isolates, of which 6 populations were from Southeast Asia and 1 from South Africa. And it's important to note here that we've got 2 populations from Indonesia. All of our populations were geographically separated from one another, and you can consider them as independent populations. The size of our pie chart is proportional to the sample size used. So we found 38 genotypes in our collection, of which 37 were limited to their specific population, and only 1 genotype was shared among some of our populations. These were some isolates from Thailand, all of our isolates from Vietnam and South Africa, and 2 isolates from North Sumatra and Indonesia. These were coincidentally the two isolates that is the oldest in our collection, and they were sampled during the original disease report from 1995. If individuals are regularly moved between populations, we would expect more genotypes to be shared among our populations. For Malaysia, we had 20 genotypes, which was by far the most genotypes in any population. This was very surprising because this is the most recent report of destructans. We would expect that older populations would have more genotypes. And we can see this in our 2 oldest populations, where they had the 2nd and 3rd most genotypes. When you compare it to the youngest population, or one of the youngest populations from South Africa, which only had 1 genotype, and that was completely clonal. If we separate our genotypes according to their Bravo genetic distance in a minimum spanning network, they generally cluster according to the geographical location. And we can see for the Malaysian genotypes, they did not intersperse with any of the other localities, and they were separated from the other populations by larger genetic distances than we typically found. So you can especially see that here at the bottom, for the South Sumatra green genotypes, they were much closer together compared to our pink genotypes in Malaysia. We can also see that the 2 Malaysian populations' genotypes also cluster close together. When we assessed the population structure, we used 2 different methods, and they gave us the exact same result. We found that the populations clustered in 4 different clusters, and they were generally according to their geographical region. The 2 Indonesian populations formed in cluster 1. Populations with a shared genotype was in cluster 2. Isolates from Malaysia in cluster 3, and isolates from China in cluster 4. We can also see here that the Malaysian population had the highest levels of admixture, specifically with those from South Africa, Thailand, and Vietnam cluster, as well as from the Chinese cluster. So we would expect that the Malaysian cluster would have the most or the highest genotypic diversity. And this was confirmed when we test for the genetic diversity. We found that Malaysian population had the highest genetic diversity of 20%, which was nearly double that of South Sumatra, which was one of the older populations and our second most diverse population. So you can see all of the other populations had really low genetic diversities, and this is because we found a really high number of clones in all of our data sets. So next we assessed the mating type distribution. So we found that Teratosphaeria destructans uses an outcrossing mating strategy. What this basically means is that we need two individuals of opposite mating types for sexual reproduction to occur. So we developed mating type specific primers in the conserved regions of these genes, and we used these primers to assess the mating type distribution. So we found for all of the populations except for Malaysia, the ratio of mating types did significantly deviate from the expected 1-to-1 ratio, which indicates that asexual reproduction is the dominant form of reproduction, and sexual reproduction is highly unlikely to occur. For our Malaysian population, this distribution did not significantly deviate from the expected 1-to-1 ratio, indicating that sexual reproduction is likely occurring. And we also confirmed these results by assessing a random recombination of our full and clone-corrected data set of Malaysia. And this showed the exact same result, that sexual reproduction is likely occurring. For the South African population, as previously mentioned, we had one genotype, so that was completely clonal. But we can also confirm here that we've got one mating type. This just confirms our previous thought that the pathogen was introduced into the country from a single introduction, and that there's not continuous introductions into South Africa. So if we look at our reproduction strategies, for populations from Indonesia, Thailand, South Africa, and Vietnam, the asexual reproduction strategy is clearly the dominant mode. This is because we found low genetic diversities, skewed distribution of mating types, no to little evidence of recombination, and a large number of clones. For the Malaysian population, we've also seen a lot of clones, which indicates that asexual reproduction is also occurring in this population. It is not that surprising that asexual reproduction is occurring in all of our populations, because the asexual state is regularly found in heavily diseased plantations, and this is also the only state that has ever been found in the field. So Malaysia is the only population where we found evidence of sexual reproduction. This is because we found it had the highest genetic diversity, it had an even distribution of mating types, as well as evidence of recombination. This was very surprising because it contradicted everything we've known for other Teratosphaeria destructans populations, especially those that's been older populations that's been more established in the area, as well as other Teratosphaeria species that's closely related to this pathogen. They all appeared to only reproduce asexually. So sexually driven evolution of destructans in Malaysia is very concerning, because it may lead to a more adaptive pathogen, which is less responsive to disease management practices. So the big question remains, what is the origin of this pathogen? So we believe that native populations of fungal pathogens typically have high genetic diversities compared to their introduced counterparts. This is because introduced populations usually emerge from a small number of individuals, whereas native populations have had long periods of time to evolve. So does this mean that Malaysia is the origin of destructans? No. It is unlikely that Malaysia represents the origin, because the pathogen is specific to eucalyptus, and eucalyptus is not native to Malaysia. To add more to this, the first plantation, eucalyptus plantation, established in Malaysia has only been in 2008. So they've got a very young eucalyptus plantation industry. So it's safe to say that all of our investigated populations represents non-native populations, and the origin of this pathogen remains unknown. But it is believed that either Australia or East Timor is the origin. This is because the majority of eucalyptus species are native to Australia, and other Teratosphaeria foliar species were first observed outside of its native region before it was finally found in Australia, and this could also be the case for destructans. For East Timor, destructans has been reported to cause disease in East Timor on one of their native hosts. Unfortunately, cultures were not available for us to test. So if we look at the movement of pathogens, we found strong geographical structure, which suggest independent introductions into all of our samples areas from an unknown source. However, North Sumatra did share a genotype with isolates from South Africa, Thailand, and Vietnam, and it is probable that North Sumatra could have been the source for disease outbreak for at least one of these countries. And then lastly, North Sumatra and South Sumatran genotypes were very closely related, and it is likely that the disease outbreak in South Sumatra came from North Sumatra. So to conclude, the long-distance spread of destructans between countries is clearly a consequence of human-mediated movement of especially infected plant material. This highlights the need for much stricter quarantine measures when plant materials moved to established plantations, and care must be taken to prevent further spread. I would like to thank everybody that is involved in this project, especially our funding bodies, for their invaluable contribution. If you would like to know more about our work, I will be available for questions. You can also contact me, and we did publish our findings. Thank you all for listening.